All right, so when we're talking about isomers, let's talk about what we mean by isomers. Isomers are molecules that have the same formula, but they're slightly different. Even though they have the same number of carbons, same number of hydrogens, same number of oxygens, how these atoms are bonded, what orientation, what angle you put them together, at what angle you put them together makes a difference in the molecule. And because there's a difference in the molecule, there's a difference in its chemical properties. So you got to understand that isomers can be very, very similar. Optical isomers can be very, very similar. And in fact, here's an example of an optical isomer. We call them mirror images. Is everybody aware that if you have writing on your shirt, and you look in the mirror, have you paid attention, do you know that the writing is backwards in the mirror?
Yeah.
All right. That's what they mean by mirror images. If you look at the drawing, you'll see that OH and H here are reversed. They're backwards. That's what makes beta glucose and alpha glucose. They're both glucose. All the carbons and hydrogens are connected the same way, except that the OH and the H are inversed. They're just flipped. That little flip changes them slightly. That little flip changes them slightly. This is one of the reasons why, and we'll see when we get into complex carbohydrates in a minute, when we get into polymers of carbohydrates. This is one of the reasons why that chair you're sitting on doesn't dissolve in water easily. It's just simply the OH and the H are in different places. That difference makes a big difference overall. But they're both C6H12O6. There's another C6H12O6. It looks something like this. Where have you seen something like this before?
DNA, yeah.
It looks like it looks like a pentose, doesn't it? Except that it's not, because it has this added thing here. And what does that mean? How many carbons does it have? Count them. Everyone look at that and count it, please. You have to be able to identify these on tests. This is how it will be on the next test. So again, we're sitting here. Let's redraw this to make sure we get this. So it's not 5. How many carbons do I have why did it do that? That's weird. How many carbons do I have if I do something like this? What do I see? What do you see here? So every time the lines connect, there's a carbon is correct. So what do we have? 6. Should we count them?
No.
1 prime, 2 prime, 3 prime, 4 prime, 5 prime, 6 prime. 6 carbons. 6 carbons. Let me see them. What kind of covalent bond? Glycosidic bond. It's a glycosidic bond is the bond that holds these together. So let's review. Let's make sure you understand. A bunch of amino acids connected together. They're connected together by peptide bonds, so we call them polypeptide.
Wait, you access instead of...
What?
Yeah, you access instead of telling us.
Oh, all right. My bad.
Oh, yeah,
Well, you should have in your notes. It's all in the videos. So when we connect, we can connect DNA nucleotides together, right? Using what group connects one nucleotide to the next nucleotide in covalent bonds in DNA and RNA?
Polypeptide.
Now, hydrogen bonds connect the 2 strands together. What connects one nucleotide to the next? What connects the A to the T to the C to the G? There's a molecule.
Polypeptide.
No. You gotta straighten that out, right? It's a big misconception. You gotta put on
I
index
got it on my notes
cards.
though.
Yeah, but it's not gonna help you on a test. You gotta put them on index cards, you gotta practice. So in between nucleotides, you have something called the phosphate. You have a sugar phosphate backbone. You remember that?
Yeah.
Okay. Well, this is a connection here. These two can be connected. Again, by using dehydration synthesis, you can connect these two covalently to an oxygen, right? And if you connect them covalently using an oxygen, we call these two we call this sucrose. And sucrose is, you know it as table sugar. So when you eat table sugar, it's a mixture of fructose, it's a disaccharide. The word saccharide, do you know what it means?
Saccharide.
Saccharide means sugar.
Oh, yeah. Oh.
These are vocabulary words, you gotta know them, you gotta be able to use them.
Because monosaccharide. Yeah, they say that in the video.
Monosaccharide is one single sugar. So this is a monosaccharide, glucose is a monosaccharide, fructose is a monosaccharide. Together they form sucrose, which is a disaccharide. So now we're up to two. Obviously we're going to make long chains. We'll talk about those in a minute. So let's go down the list. We talked about optical isomers and what they are. This is a structural isomer. Fructose and glucose are structural isomers. They're arranged differently. Substantial arrangement, right? The carbons are connected differently. Do you agree? They're both C6H12O6, but they're connected differently. So this forms a pentagon. This is a hexagon, right? It's 5-sided. This is 6-sided. But this is still a hexose. This is still 6-carbon sugar. And as you saw, the difference between the optical isomers is a slight difference in arrangement of orientation. The oxygen is still connected to this carbon. That oxygen is still connected to this carbon. Is that OH group up or down? That's the only difference. All right. So sucrose is a disaccharide, and the difference between optical and structural isomers, I think we've gone over. I hope you understand. Do you have any questions? Is there anybody that's confused about this?
Yeah, you said what is this?
All right, so we have two different kinds of isomers, right? Optical isomers, which is you're gonna have to do some homework. I already said it. Go back at the beginning of this video and review it. Okay. So isomers are two molecules, and by the way, the definition is in the reading that you should have done already. So they have the same formulas, right? Same structural formulas, but they're different in how you put them together, right? They're slightly different. So there's two kinds of isomers that we've talked about thus far. There's an optical isomer. Iso means the same, right? So there's two that have the same formulas, but they're slightly different in which way they're pointing. They look like they're backwards, right? Mirror images of each other. Mirror images. Optical means sight, right? Vision, like you go to an optometrist for your glasses. So an optical isomer is an isomer that just looks different. They're mirror images of each other. A structural isomer is actually when they've been rearranged. The carbons have been rearranged. So I hope that it's easy for you to see that this is 6-sided and this is 5-sided. I hope that you see you have the same number of carbons, you just connected them differently. And because you connected them differently, you got fructose, and over here you have glucose. Put them together and you get sucrose. Sucrose. Table sugar. All right, sucrose. OSE means sugar. OSE means sugar. So if you see sucrose, fructose, glucose, galactose, lactose, all those are sugars. Disaccharide means there's two monomers of sugars. So you got to make sure you put those on index cards or whatever you got to do to memorize that. You got a question?
So the sugars are connected together by oxygen?
That's correct. It's called the glycosidic bond. Why is that? Because what had to leave? How do we put things together? Water had to leave, is correct. So take a look at it. An OH and an H come together, and what comes out? H2O, water,
Water, hydrogen bond.
right? When that happens, when we remove water, what do we do with that after this leaves, right? Oops. Should do it this way. After the water leaves, what are you left with? The oxygen's left over, and that connects the two monosaccharides together.
Okay.
That gets over and over and over and over again. It's just like peptide bonds. It's just like DNA and RNA. All of them do the same thing. It's called condensation reaction, or called condensation reactions, or dehydration synthesis. Or dehydration synthesis. Is that okay?
Yep. Understand.
All right. And I know you understand because I can hear you answering questions, even though some of you are doing it under your breath. I see you nodding. There's at least one student who has their back to me pretty much the entire period. So other than that one person, I'm sure their parents and I will have long discussions on that. I see that you're getting it here and now. That's great. But you need to know it on the test. Which means you have to take it to the next level. You have to be able to put it on index cards and memorize it, quiz each other so that you get it. And I know some of you quizzed each other for last quiz. You got to figure out what you did wrong. Make sure you understand what's going on and what's important. I told you what's important here. Dehydration synthesis, right? Or condensation reactions, glycosidic bonds. What's an optical and structural isomer? The fact that we can connect them together with these glycosidic bonds to form polymers. Monomers of sugar are what? What do we call monomers of carbohydrates?
Glucose
Not lipids. Lipids have no polymers or monomers. Somebody said it.
and glucose and
Glucose and fructose are monomers of carbohydrates. That's correct. We also call them in general monosaccharides. Monosaccharides. Right. Now, what is the monomer of proteins?
What is it, monomers of proteins?
Amino acids. What connects the monomers together into a polymer of proteins? In proteins, what connects the amino acids together? What kinds of bonds? They are covalent bonds, specific. They have a name. What are they? Peptide bonds. When you have more than one peptide bond connecting a bunch together, what do you call that polymer? Polypeptide. See, do you have that chart? Did you create a chart? Are you guys able to sit at lunch and write that down without question? Because if you're not able to do that, if you're getting stuff confused, then you're going to get it wrong on the quiz. No matter how much, and I know you understand it right here when I say it, because you're like, oh yeah, I remember. Yeah, but I'm not going to be able to help you on the test or the quiz. All right.